We have a department YouTube channel, and with this we are reading this camera, so, going to all the participants. And I'd like to inform one participant from France. So, so for 10:30, it is 7 o'clock your country, so try to manage this. Okay, I do it. Generally, because we stay in the night. So see, join this when to international level. I from Karnataka, and to within 12 days. So this is that the students also understanding the importance of these advanced biology technique. This is about you learn, like your last time the left one will come to know. And if you have any kind of query, any kind of question, don't hesitate to ask. Now, this is said that I should inform you about the biotechnology department research ongoing. So with this system, it is easy to understand what research we are doing. Basically, we are doing the genetic engineering of crop plants and proteomics kind of thing. We have a nanoparticle plant research. In fact, just like our university department, we are also writing project, writing research paper, the patent also. My students are also qualifying other national examination, and now it is the number is increasing. Right? So this is all our PhD. But I remember in 2015, Professor was here, and he was roaming around for the preparation of NAC. At that time, we were facing some kind of trouble. Our face was very down. He come. He says, "See, nobody will ask for 10 years. What problem you face? Will ask what you did. How do you potential doing?" We have we have like company, but we use the chemistry, and now that this is like 10,000 rupees you can make like half liter, and we have given this RNA buffer to various companies also, like High Media. They have also taken this, various laboratory. This is equally good for the plant, for the cell, for the insect. So you can isolate RNA very quickly. In fact, my students, PG students, they used to prepare this buffer ready and used to isolate. So the research is, RNA isolation process is cheaper. So this is, I understand that this is a kind of achievement, and we name this as a tetra AP, because tetra is 4 compound mixed together. AP is my student sake part. So I just dedicated to it. It's a very hard working. The second product that very important I should inform, because we are guided in that research, we have developed nanoparticle based herbicide, and the news also flashes. We have submitted the patent also. Basically, this is an organic herbicide which is equally effective as chemical herbicide, and we have distributed to several villages. In fact, we have also tested. This is what news flashes in national TV. So I got a call from IFO, some PR. He said that we will be visiting your department, and we want to see. I hope that this will go, and I'm very sure that this will come to the market also. The second thing that we are doing, since Professor sitting, and I'm very happy to tell that in fact I'm the single faculty. In fact, my colleague will be joining very, but somebody should be there. But what we are doing is we have developed PDF file, they have developed protocol, and the second will be a YouTube video through our YouTube channel, department YouTube channel. So before going to the lab, we will see all the steps, how it is being done. In fact, several titles we have already made it. But then before the lab, kind of thing, our students will certainly get this benefit. Other department can also make this kind of videos, so that you should not be present every time in the, all 4 hours in the laboratory. And in fact, this I made to save my time, but this is working very fine. So and since all the students have mobile, and this is not like our time that see it, believe it. Now this new generation, they say see at the screen and then believe it. So they see it, they see it, and then believe it, and they do it also. So this is the thing that we are doing, and I am sure that we will do even better. We have certain other kind of thing, but this is not the forum for to discuss. Certainly, if somebody is interested, so can, because we have recently started the EV also, and a very good result is going to come. So thank you very much. This is all about the workshop and the department activities. So thank you very much.
Always about to we heard a lot about class from our professor faculty in the department. We know a word professor among the top 2% of the scientists. I would like to request the organizing secretary, Dr. R.K. Koiri from Department to give the details of class.
Thank you, Aditi. Famous and is more than any introduction. सर शायद अगर मैसेज होगा तो प्रोफेसर व्यास सर के बारे में ही जानता हूँ. But as the custom goes, I would like to take the privilege to introduce Professor Vyas sir. Professor Vyas is a professor in the political sciences. Dr. Singh Vyas about years of teaching and experience at the postgraduate level. He is a pioneer scientist in the field of nanobiotechnology and immunology. He has judicially exploited bioligand for targeting bioactive antigens. And 100 students. Published in journals of in fact, he has authored two reference books including pharmaceutical biotechnology, liposomal therapeutics, novel control delivery system, targeted delivery system, theory and practice, novel drug delivery system, conducted many chapters, and also on the university, such as acting vice chancellor of Dr. Hasim Vidyalayar, Dean Faculty of Technology, Head of Pharmaceuticals and Department of Technology, Chairman of the International Pharmaceutical recent patents on delivery formulation, and others. and first in India for research. Citations include 18 search for index is 240. His area of research includes developing nanocarriers for site
for a lecture.
Please.
Honorable Dean of School of Biological Sciences, Dean Chemical Sciences, Head Department of Pharmacy, Professor Kasav, Professor Das, Dr. Bhargav, Dr. Koeri, and Dr. T. Mohan, many more. सभी का नाम लूँ, but I got bisected after hearing about myself and from the mouth of your dean, respected Dr. Varsa Sharma. And after hearing about myself from Koeri Shah, so I just to my talk with this one poem. कि आईना ये तो बताता है कि मैं कौन हूँ, आईना ये तो बताता हूँ कि मैं कौन हूँ, पर इस बात हमेशा मौन रहता है इस बात पर वो रहता है हमेशा मौन जब मैं पूछता हूँ कि मुझे कि जब मैंने पहली बार देखा सृष्टि और प्रकृति को न जाने ही अनजाने में उसका हो लिया। जब मैंने पहली बार देखा सृष्टि और प्रकृति को तो ना जाने अनजाने ही मैं उसका हो लिया, और दुनिया की तो राम जाने, पर मेरे लिए तब से आज तक मेरा बुध मंगलवार की बात होता है, मेरा बुध मंगलवार की बात होता है। इट मीन्स more we move towards the nature, more you learn about the things. You may realize and appreciate that the Nobel Prize in 2022 that went in the chemistry experiment, more we a person towards the who discovered Greek chemistry, more you learn and about Greek chemistry is nothing else. That is the only the natural way appreciate the chemistry, that is the biochemistry at large, and what nature is doing, and just modern the environment all around and the ambience for the survival, who discovered Greek chemistry. Likewise, let me put a bold after the invention of new generation sequencer, it was possible in 2040 that Jimmy Carter was diagnosed cancer of multiple organs. Jimmy Carter. This is really exciting. I am feeling elated. He was diagnosed and he came to public saying that probably I am for a few days to be with you because my life is going to be claimed by dreaded disease like cancer. New generation sequencer that came to rescue and the cure pharmaceuticals. They just adopted Jimmy Carter as a patient, and personalized medicine was developed. What was that? That using that CRISPR Cas9 editor, they could conjure a receptor on the surface of B lymphocytes. Just be careful listening me. They conjured this expressed that particular receptor that is chimeric in nature, so that all type of anti simply caught about. So What was first? That is B lymphocytes from the Jimmy Carter blood, which were modified with chimeric type, that is CAR receptor. So these lymphocytes are referred to as T lymphocytes CAR. And secondly it was seen that what are the genes which are aberrant within this particular patient. They were found to be CD1 checkpoint, and another was CTLA4 antibodies, which are just suppressing our own immune response. Let me once again share one word with you, that in our lifetime there is no even a single individual as a survivor who is not developing cancer within the body. Everyone is developing cancer within the body, but our own immune system, it just repairs it. It takes care of, and this happens normally. But in case of cancer, when it develops to its notorious stage, then it is the story. So Jimmy Carter, he was detected with this. So what was done? That was in 2014, October, he was detected with the cancer. In December, he received first this therapy that was based on CAR-T lymphocytes that was injected, along with antibody to CTLA4 and CD1. CD1, what is CD1? That our biodrones, these are biodrones, means our B lymphocytes, T lymphocytes, they are biodrones, they are spanning all entire body and searching for anything which is nonsense to the body. They search, shoot at the site and eliminate from the body. This is how it is all the day, all the time happening within the body. So first dose was of CAR-T, modified CAR-T lymphocytes that was infused in, followed by the dose that is antibody against CD1. What is CD1? CD1 is the call from the cell that let me live. I am from your own condominium. Please don't kill me. So this is the call. Always the cancer cell, they raise as a voice that we shouldn't be killed like, and they escape our immune surveillance. And CTLA, which is our activated system, is getting suppressed by, when once this checkpoint is broken, then probably they come to the activated form. So first dose was in October 2014. Second dose was given in May 2015, and third dose was given in October once again in 2015, and he was declared free of any cancer symptoms or any cancer. Let me again share one word, like all chemotherapy, radiation and other things, they minimize this carcinoma, but they cannot cure carcinoma because of the basal cells which are left out. Mesenchymal cells which are responsible for the growth of the cancer. And can you believe it? After he was under the hospice care, that is the care given by the psychologist, the care given by this counseling purpose, and also the physicians, nursings, and the rituals, the religious peoples all around, he could complete 100 years this October 1st and still alive a person. What could be more this exciting news as compared to this? It means that if we are near the science, we are more into the science, we dive more deep into the science, probably we can unravel and mine the mystic and mysteries which can be taken to the benefit of science and also to the common, to humankind, and also to the betterment of this world. Let me see that what is important at core. I always feel jealous of you because you are better updated than me. The available this digital platform, the available instrumentation facilities with you, and also your inclusive, tenacious involvement with all endurance adherent to it in the science, that you are the passion and love to the science, and you are dedicated to the science, and you are taking it to the furtherance. So science is getting benefited by, community is getting benefited by, and you yourself is getting benefited by. This is something great about. When you feel that what you are doing, when you feel what you are, then probably I think that this life is meaningful. मैं एक और कुछ आपके लिए लिख के लाया था, उसको मैं जस्ट देख भी याद तो होगा मुझे कि कैसी अजीब शर्त है दीदार की, किसी को देखने की कैसी अजीब शर्त है दीदार की कि जब आंखें बंद होती है तो जलवा दिखाई देता है, जब आंख बंद होती है तो जलवा दिखाई देता है। यदि आपको जो देखना है एक्चुअली व्हाट यू आर और व्हाट यू कैन बी, देन आपको ये ज़रूरी है कि आंख बंद करके अपने खुद में ही आपको जाना होगा। एंड एक आई डोंट हैव द पैरेलल वर्ड्स अवेलेबल इन द इंग्लिश डिक्शनरी। अजस्र, ज्ञान का अजस्र प्रकाश जिसे कहा जाता है, मुझे यहाँ नहीं मिलता है कि इंग्लिश में इसका क्या पैरेलल होगा, लेकिन कोई सोच सकता है कि जब कोई लौट के वहाँ जाता है, द सेरेनिटी एंड द कंटेंशन and the total satisfaction that it derived, it is beyond the words for the expression. It is something really, one cannot imagine of it. What that is, it is mesmerizing. So more science you do, more you will be with you, more you will be, your this sanctity, your veracity will get verified by your involvement in the science. Your work will be axiomic by and epitomic by character for its disposition. It will be perceived, pervasive in nature. If it is pervasive in nature, probably your presence is ubiquitous. You will be known everywhere, every nook and corner. You will be known to yourself and known to the community all around. जब दुनिया आपकी ओर देखे कि आपके होने से उनका होना है, तब आपका होना सार्थक है। जीवन में सफल तो बहुत हो जाते हैं लोग, लेकिन सार्थक होना बहुत विरल है। आई एम रिपीटिंग दिस, जीवन के होने के मायने सफलता नहीं है, जीवन के होने के मायने सार्थक होना है। यदि आप सार्थक हो सकते हैं, प्रोबेबली हेड्स ऑफ टू यू। आई एम जस्ट रिविजिटिंग माई डे व्हेन आई वाज हेड ऑफ द डिपार्टमेंट। इट वाज ए barren land, like only name and the poster in the name of biotechnology was there. But after the incumbents of Dr. Upadhyay, Dr. Anupam and Akanksha, अनामिका, तुम अब थोड़ा ना, यदि मैंने नाम नहीं भूला तो मैं जवान नहीं बना रहूँगा, इसका भी तो मजा लेने दो। भूलने में जो मजा है, और जब आप सबके साथ किसी को याद किया जाए तो आप क्या जानो उसका सुख क्या होता है। किससे कहूँ मैं, किससे कहूँ पीर अपने पिया की मायरी, अपने जिया की मायरी। इस चीज तो वो जाने जिसको जो जिया जाता है। भूल के किसी को याद करना, कभी ऐसा होता है जो याद आ जाए उसको भूला जा सकता है क्या? लेकिन सायास किसी को बुला के फिर याद किया जाए, उसका होने का कुछ मायने हुआ करती है। इनके आने के बाद आफ्टर देयर इनकमेंस इन द रिक्रूटमेंट, आई हैव सीन lot of difference, and heads up to Dr. Upadhyay that single-handed, a single-man army, what he is doing, and he could simply fetch a number of extramural funded this research projects on his own, and he just deployed the funds and the simply structuring the department to the conform where it should be otherwise. It's great. It's great. And can you believe he is inclusively involved in the teaching, in the workshop, and what he was just referring to and making a mention of that barcode track of every instrument is available in the department. Who worked on that particular instrument and what was the output of those particular instruments and how the things they are going, what are the protocols being developed, everything there on the fingertips. So updation of this department, it is beyond my imagination. And मैं इसको कहने में बड़ा गर्व फील करता हूँ कि बरुआरी अखियन नरक अखियन को सुख होत, बाप बेटी से हारे, और शिक्षक अपने विद्यार्थी हारे, इससे बड़ी जीत दुनिया में होती क्या है? इससे बड़ी भी कोई जीत होती है क्या? I haven't seen, I cannot realize that any victory can be even greater than this. युद्ध के मायने ये नहीं होती कि दुनिया मिटा दी जाए, युद्ध के मायने ये होती है कि दुनिया परिवर्तित कर दी जाए। युद्ध लड़ना हो तो परिवर्तन के लिए युद्ध लड़े जाएं, ना कि दुनिया को विनाश करने के लिए युद्ध किए जाएं। मैं आपसे ये बात कर रहा था कि रियल टाइम क्वांटिटेटिव PCR and new generation sequencing systems, that is the advanced version of the Sanger technology, though it still remains to be the gold standard in sequencing. The Sanger one, all the steps they are covered together, and what he is referring to that both the end, otherwise one end, this synthesis it normally proceeds within the biology, but both the ends they are just moving towards and meeting together, and at the same time that is connected, it is interfacing with the sensors, so that AI and others they can simply monitor the progress and what type of this nucleotides is getting attached to that for complementation. It is signaling out, and if anything odd in nature, it is with the different color composition, it is seen on the that particular screen or monitor. This is something really great. SNPs, they have got great role in the single nucleotide variation or polymorphism, what we are referring to. It is of inseminal. It is inseminally instrumental in detecting the and diagnosis of the diseases of different types, because single this SNP is responsible for so many diseases. And beyond it, I'm just adding another word like the Alzheimer, the disease, that what we are forgetting. Can you believe it? A day it won't be simply there on this earth surface. The way the research is progressing, what that research is, that is based on that sequencing and also the CRISPR Cas9 editing system, which can silence the gene responsible for, which can also even what we call it, the siRNA can be simply synthesized for the purpose of silencing of aberrant this expression of that particular amyloid proteins. If if it is possible that we can at the early stage itself we can detect that one individual is having the possibility of developing Alzheimer at the later age, and that can be taken care of with the help of siRNA or microRNA, or even the editing of the gene which is gone early and notorious as far as expression for the benefit of this biology is concerned. मैं ये कुछ एक बार और देख लूँ, कुछ अच्छी चीज़ आपके लिए मैं लाया हूँ। तो प्राय मैंने इसमें से जो हिंदी की बातें थी आपसे कह दी। कहने की तो बहुत सारी बातें हैं, कहने को तो बहुत सारी, यदि हम साइंस में ही जाने लगे, लेकिन मैं इस बात को मानता हूँ कि आप बड़े सौभाग्यशाली हैं कि आपकी समय में ऑल दिस फैसिलिटीज इंफ्रास्ट्रक्चर बाय नेचर दे आर अवेलेबल। एंड एक और बहुत बड़ी बात है कि शायद तो नहीं कहूँगा मैं फॉर श्योर दैट यू आर लिविंग इन एन एरा, you are living an era with all facilities and opportunities to work in the science available with you, that you can directly enter into the circuitry of inference, where no enigma, no divides, no confusion, no all types of beings they prevail anymore. So if you are sure about all these things, that what you are getting, they are verified, they are this sanctity and their truthness, then probably you are the strongest person who can work more and more rigorously in the science, more and more rigorously in the science. And working in the, ये ऐसा एक रास्ता है, एक ऐसी प्यार की ये दुनिया है जिनमें देखिए, ऐसा दुनिया में कोई भी मनुष्य पैदा नहीं होता जिसमें द इंस्टिंक्ट ऑफ लव होती नहीं होती, लेकिन सबसे बड़ा प्यार ये है व्हेन यू स्टार्ट लविंग बायोलॉजी। जब अपने से प्यार करने लगो, इतना प्यार करने लगो, आप ऐसी दुनिया है कि जहाँ तुम अपने प्रियतम से कभी बिछड़ते ही नहीं, राधा तो इंतजार में पूरी जिंदगी सूखती रही, लेकिन तो यदि आप प्रेयसी बन जाती है इस प्रियतम की जो बायोलॉजी है, तो ये सौभाग्य है कि सदैव आपको मिला रहती है। नहीं समय मैंने कहा ना, जब जब आँख बंद करोगे तो जलवे होंगी, और ऐसा करते करते यदि आप करते रहे तो मैं आई एम मोर देन श्योर कि जब जब जलवों की बात होती है तो लोग तुम्हें याद किया करते हैं, लोग तुम्हें याद किया करेंगे कि आपने ऐसे जलवे बिखेरे हैं, ऐसी चीज़ों को जाना है। एक और चीज़ आपसे कहना चाहता हूँ कि द इनकल्केशन ऑफ द नॉलेज, इससे बड़ा ट्रेजर दुनिया में कोई नहीं है। नो ट्रेजर इज मोर देन दिस पर्टिकुलर ट्रेजर। इसको हम शायद जब मैं वृद्ध होने लगा 71, आई एम कंप्लीटिंग 72 और मूविंग इनटू, तब मुझे ये लगा कि मैंने अपने दिन ऐसे ही खो दिए, ऐसे ही खो दिए। ज्ञान कितना असीम है, कितना अपार है, इसकी आप ये समझिए कि आइंस्टीन ने एक बहुत बड़ी बात की। आइंस्टीन जो इस दुनिया का सबसे बड़ा विद्वान पैदा हुआ हमारे समय का, तो उसने एक बहुत बड़ी बात कही कि मेरी जिंदगी की केवल एक उपलब्धि है, केवल एक उपलब्धि है कि पूरी जिंदगी ज्ञान अर्जन में खपा देने के बाद मुझे एहसास हुआ कि मुझे कुछ भी नहीं आता, मुझे कुछ भी नहीं आता। अब सोचिए, आप जब पूरा ज्ञान व्यक्ति को हो जाता है तो आप कई पर्दों के परछिनों की पार चीज़ों को देखते हैं। बस एक एग्जाम्पल के साथ मैं कहना चाहता हूँ ज्ञान की ताकत होती क्या है। पॉल एहरलिच, उसको 2 बार नोबेल प्राइज दी गई, 1902 में तो उसकी बार मिली। उस समय कोई इक्विपमेंट्स, ना इलेक्ट्रॉन माइक्रोस्कोप था और ना कोई आपकी qRT-PCR एंड नथिंग वाज अवेलेबल, लेकिन वो आंख बंद कर लेता और खुद में विचरण करता, और उसने देखा कि क्या है कि लोग बीमार होते हैं और ठीक हो जाते हैं, बीमार इन्फेक्शन होता है और ठीक हो जाते हैं। उसने पहली बार ये सोचा, ये 1894 में पहली बार, और 1902 में उसने प्रपोज किया। क्या प्रपोज किया कि कोई माने या ना माने, कोई माने या ना माने, इस शरीर में कुछ ऐसा घटता है जो कि लास्ट काम करती है। इस तरीके से चीज़ों को उसमें से पूरा ढूंढ के उसको ख़त्म करके और हमें पुनः जीवित करने की उसमें ताकत है। वो कुछ नहीं है ये, और वो क्या होगी कि जो ऐसी फॉरेन बॉडी के अगेंस्ट लड़ रही होगी, तो फर्स्ट ही नेम देम एंटीबॉडीज, उनको कहा एंटीबॉडीज, और उनको क्या माना एंटीबॉडीज में। दुनिया में मैं एक और बात कहूँ कि इतना बड़ा प्यार का कोई कृष्ण और राधा का भी उदाहरण इससे बड़ा नहीं है। मैडम सूर्यवंशी जी है ना, दुर्गवंशी सूर्यवंशी सब देखो, जो दुर्ग का वंशी होगा वो तो सूर्यवंशी होना ही होगा। मैं तो आपको भगवान राम का वंशज बना रहा हूँ। तो दुर्गवंशी जी, मैं इस बात को कहना चाहूँ, दुनिया में इससे बड़ी कोई मिसाइल नहीं है कि आप हजारों करोड़ों जितने भी तुम्हारे मन में आए उतने प्रोटीन कॉकटेल में एक एंटीबॉडी डाल दो, तो वो कहाँ जाएगी? एंटीबॉडी डालोगे तो वो ढूंढ के कहाँ जाएगी? एंटीजन के पास। एंटीजन जब मिल जाते हैं तो कहते हैं कि अब मरेंगे तो साथ, फिर उन्हें कोई अलग कर ही नहीं सकता। मतलब आप क्या समझे कहूँ मैं इन बातों को, यू जस्ट थिंक अबाउट दिस, उनको कोई नहीं अलग कर सकता। आपके जितने भी एलाइजा मेथड्स एंड अदर्स टारगेटिंग विद इन द बॉडी, वी आर ऑल बेस्ड ऑन दिस। तो पॉल एहरलिच ने 1902 में देखा कि प्यार की वो मिसाल कि जिसके हम जस्ट पीछे हो ले, तो एक दिन हमें वो जरूर मिलेगा कि वी शैल बी एबल टू डेवलप द मेडिसिन और मेडिकेशन व्हिच आर टारगेटेड इन नेचर। टारगेटेड इन नेचर क्या है? कुछ इटियोलॉजी की व्हिच इज क्राइंग फॉर द रेस्क्यू, only for that destination. If drug can be delivered at right time, in right quantity, at right etiological destination, probably we are exempted of all possible contraindicative manifestations. कि उन्होंने देखा 1902 में, जिसके पास कुछ नहीं था, केवल बंद आंखें करने देखने के बाद उस जलवा देखने की उसने ताकत पैदा कर ली थी। और 1985 में मिलर ने एंटीबॉडीज को जब फिर से देखा तो पोस्टमसली उसको फिर से एक बार याद किया गया नोबेल प्राइज के लिए। किसको? पॉल ऑरेलिच को। तो जब हम अपने खुद के साथ होते हैं, या हम खुद को प्यार करने लगते हैं, या हम प्यार की परिभाषाएं जानने लगते हैं। मैंने आपसे कहा राधा कृष्ण मिल ना सके, सीरी फरहाद मिल ना सके, लेकिन दुनिया की भीड़ में अपने को ढूंढ लेने की ताकत केवल है तो एंटीबॉडी में है। और उसका कॉम्प्लीमेंट्री पार्ट कौन है? एंटीजन है। लेकिन हमने जाने अनजाने शब्द दे दिया एंटी। एंटी नहीं है, दे आर लवर ऑफ ईच अदर। प्यार की वो कहानी जिसके बाद वो कभी दुनिया में कोई उनको अलग नहीं कर सका, उनके रास्ते पे चल के, जिनके पथ पे चलने के बाद चीज़ों के समाधान मिलने लगी। मैंने अपनी एक बात कही थी मैडम कि रामायण, या लोग कहते हैं तो रामायण में और कुछ मत देखो, राम के वो चरित्र के जीवन का चलता हुआ पथ जिस पे चल जाने के बाद मनुष्य में जो पशु की तरह माना जाता है उसमें दिव्य हो जाने की संभावना बन जाती है। उसमें ये छलांग मारने की ताकत आ जाती कि वो दिव्य हो जाए, और दिव्य से भी आगे जाके वो हो सकता है। संभावना ये भी बहुत है कि कभी भगवान हो जाए, क्योंकि जानत तुम ही हो जाइए, ये ताकत है उसमें। तो ये पॉल ऑरेलिच ने आज की एक शताब्दी पूर्व, जब हम वी आर स्ट्रगलिंग जस्ट फॉर इट्स टोटल फ्यूजन, दैट इज द टेक्नोलॉजी या द हाइपोथेसिस बेस्ड ऑन दिस व्हाट वी कॉल इट, बुलेट, मैजिक बुलेट। तो उसके वो आज जिसको उसने तब देखा, कैसे आंख बंद कर ली, वो मैंने कहा ना कि कैसे अजब से शर्तें हैं इस दीदार की कि आंख बंद कर लो तो जलवा दिखाई देता है।
1893 वर्नर को भी ऐसा ही हुआ था।
वर्नर, यस।
अब तो 2 घंटा बैठ के जो थ्योरी निकाली थी, बाद में पूरी केमिकल बैलेंसिंग उसी 20 साल बाद पता चला।
यानी इवन द कैकुली हाइपोथेसिस ऑफ द हाइड्रोकार्बन्स, दे ऑल, इनमें तो इंट्रोस्पेक्ट किया। ये तो सब इंट्रोस्पेक्शन था। दे आर जस्ट थिंकिंग एंड जस्ट दे आर लिविंग विद दैट पर्टिकुलर टाइप ऑफ द कंसेप्ट डे एंड नाइट, इवन इन द ड्रीम। यही तो है कि जो नींद लगने पर भी जो तुमसे देखो, नींद लगने के बाद वो नींद जिसमें स्वप्न ना आ जाए तो मुक्त हो गए, मरने के जैसा है। लेकिन वो नींद जो स्वप्न में भी वो चीज़ दिखाए जिसके लिए तुम जीवित हो, और जीवित में भी वो सपने दिखाए जिसके लिए तुम्हें जीना है, वो नींद नहीं होती है। हम उसको सही मायने में ही उसको चिंतन कहते हैं। चिंतन की परिभाषा ही ये कि सोता हुआ हमारा अनकॉन्शियस भी वही देखे, और जागी हुई आंखें भी वही सपना देखें। यदि इनको हम देखना शुरू कर दें तो मैं देखता हूँ कि अपार संभावनाएं। यदि मैंने खुद को भी खोजा होता तो मैं इस कहानी को क्यों शुरू किया था मैंने, जिमी कार्टर की कहानी की। Jimmy Carter, the story, it started in 1914 on the concept. The Nobel Prize was given in 1917. In 1917, I make my colleague to recall, I made the same presentation in plenary lecture of IPC, Indian Pharmaceutical Congress. Only thing is that I was lacking the confidence. They were axiomic and epitomic because of the confidence and the facilities available with them. मुझे ये नहीं मालूम था कि जो मैंने निकाल दिया ये सच में है कि नहीं, लेकिन they were more than sure कि ये तो है। और जापान के नोबेल प्राइज भी, और जिसको राजीव वर्मा ने हमारे साथ में PhD 1917 में सबमिट की थी, that was awarded and available with our library. The same concept. इसका मतलब क्या हुआ कि हमें खुद को जगाना होगा, खुद में विश्वास लाना होगा, और ये विश्वास लाने के लिए what facilities now being generated and they are available for you. You are fortuitous, you are fortunate to have this era to live. जब आपको ये सभी फैसिलिटीज मिल रही हैं, आई होप यू हैव टू गो ए वेरी वेरी लॉन्ग वे। मैं डिपार्टमेंट ऑफ बायोटेक्नोलॉजी की बात कर रहा था, जहाँ आज से बहुत दिन पहले विद meager limited facilities he developed on his own the cell culture technology and the identification of genes responsible for drought resistance under the stresses. This is what for the potato he could do it. And chia seeds, he is working for this tissue culture technology. And not only this, that this bio-compatible and eco-friendly systems for nanoparticle synthesis he is developing in the laboratory. And you see that almost all facilities now they are generated. अब मैं एक और बात कहूँ, कार की आपने बात कही, एडवांस सेंटर. आई थिंक एडवांस सेंटर that should be the center for biological sciences different, and advanced center for bona fide or basic researches it should be different. Two different centers should be there. इसको इस बात में इसलिए कहता हूँ कि this is trigger one stop solution for research problem. एक जगह पहुँचो, आपकी हर समस्या खत्म हो जाएगी. आपको न्यू जनरेशन सीक्वेंसिंग कैसे चलाना है, आपको सीपी उपाध्याय में लगे. आपको ड्रग डिलीवरी के बारे में क्या कैसे करना है, मैं अवेलेबल हूँ. आपको टिश्यू कल्चर कैसे करना है, सेकेंडरी मेटाबोलाइट्स मैनिपुलेशन मैनरिंग कैसे करना है, हमारे पाटिल साहब अवेलेबल हैं. आपको बायोलॉजिकल सिस्टम में इन सिस्टम को इनका स्टैंडर्ड फॉर्म कैसे हैं, प्रोफेसर वर्षा शर्मा इज अवेलेबल. एंड मेडिकल साइंसेज में कैसे को हम देखते हैं, डॉक्टर इज अवेलेबल. इफ ऑल अंडर द सेम अम्ब्रेला एंड सेम रूफ दे आर अवेलेबल, देन ऑल प्रॉब्लम. इससे क्या होगा, इससे हमारा एक्सपीडिशन जो इन साइंस है दैट विल गेट एक्सपेडाइट, और हम जल्दी चीज़ों को जानेंगे. और जो चीज़ें हमारे के द्वारा की जाती वी विल बी मोर कॉन्फिडेंट अबाउट देम कि ये बिल्कुल ऐसा ही है. यदि ऐसा होगा तो डेज आर नॉट फार दैट एवरी अल्टरनेट ईयर एट लीस्ट इंडिया विल बी फेचिंग वन और अदर नोबेल प्राइज इन द एरिया ऑफ साइंस. और यह संभावनाएं मैं आप सबके चेहरों में पढ़ रहा हूँ. आप आईनों में देख के संभलते रहो, मैं तुम्हारे अंतः को पढ़ के समझता रहूँ कि आपके अंदर क्या छिपा बैठा है. तो आई थिंक कि दिस इज गोइंग टू बी वेरी प्रोडक्टिव वर्कशॉप ऑफ 5 days in blended mode, offline, online, and a huge number of participants is reflective of that how significant and how instrumental this particular central technology that is now available with us, it is for all of you. And it is beneficial to the science, beneficial to the researchers, beneficial to the students, and also beneficial in turn to the country in particular. तो इन चीज़ों के आने से, ऐसे एक ही रूप पर सब चीज़ों को मिल जाने से, और ये मेरी एक बड़ी कल्पना थी. यदि आपको किसी को याद हो कि जब मैं कभी भी बात करता था तो मैं एक बात कहता कि देयर इज ए नीड ऑफ अ सेंटर फॉर बायोलॉजिकल रिसर्च. जो मैंने हमेशा इस बात को कहा था, अरे बायोकेमिस्ट की ताकत हम क्यों ना उसका उपयोग करें. मैं कैसे जान सकता हूँ उतनी सब बातों. और ह्यूमन एनाटॉमी, फिजियोलॉजी, फेनेस्टेशन कहाँ है, लीकी लेयर्स किधर हैं, कॉम्पैक्ट कंपार्टमेंट कहाँ है, मिजेनकाइमल सेल्स क्या हैं, एंड मॉर्फोजेनेसिस, एनवायरनमेंट एंड निश क्या है. मैं क्या जानूँ, मैं क्या जानूँ कि सिंड्रोम क्या है, सिम्टम्स क्या है, डिसीज क्या है, क्या है, बायोम्स. लेकिन यदि सब हमको मिलते रहे तो जो मुझे जानना है वो जानने के साथ, सबके मिलने के साथ वो समग्रता में हमें मिल जाता है, जिसके ऊपर संपूर्ण मानव जाति जिसके ऊपर गर्व कर सकती है. और मुझे गर्व है कि हमारे विश्वविद्यालय बहुत अच्छा काम हो रहा है. मेरी इसी उम्मीद है. देखो एक और चीज़ मैं आपसे कहूँ, एक ट्रेंड है जो चला जाता है, वो अपने समय को बहुत बड़ा बताना चाहता है. मैं चाहता हूँ मुझसे बड़ा वैज्ञानिक कोई नहीं हुआ होगा, वो अब तो कोई कुछ कर ही नहीं रहा. ये बिल्कुल गलत है, ये बिल्कुल गलत है. तो आप सबको बड़ी इज्जत से और आदर के साथ मैं झुक जाना चाहता हूँ कि आप में बहुत कुछ मुझसे आगे है जो शायद जिससे मैं वंचित रह गया. आने वाली जनरेशन जहाँ हम तो जहाँ बैटन आपको दे रहा है उसके आगे की रेस आप दौड़ रहे हैं. तो यू आर अहेड ऑफ अस. हम तो गई हुई जनरेशन के लोग हैं. हमारे पास क्या है देने के लिए, केवल अनुभवों के अलावा. कि यदि तुम्हारी ऊर्जा, तुम्हारे तुम्हारे उत्साह और हमारे अनुभव यदि मिल जाएं तो रावण जैसी सत्ता को वानरों के साथ मिलकर राम पराजित कर सकती है और सुंदरकांड का निर्माण हो जाता है। आओ हम विज्ञान के सुंदरकांड का निर्माण करें। तुम्हारी ताकत, तुम्हारे उत्साह, तुम्हारी ऊर्जा, तुम्हारी लगन, तुम्हारी लगातार ये विज्ञान के प्रति आपका जो लगाव है, हमारे अनुभव शायद ये कभी इंटरफेस हो जाए तो मुझे लगता है कि आसमान की ऊंचाइयां भी कम पड़ जाएंगी जहां तक हम जा सकते हैं। तो with these words that it is not destined but destination is to be decided by us, क्योंकि वो तो एक बीच का एक पड़ाव भर होगा। मैं नहीं मानता कोई कहता कि यहाँ एंड ऑफ द स्टोरी है, आप उसके भी आगे जा सकते हैं। और मैं यह सुनकर बड़ा गर्वित हूँगा कि अगले साल इस फैसिलिटीज़ के आने के बाद, जैसे ट्रांसलेशनल 2 रिसर्च की अभी बात की गई है, एक the buffer that is developed indigenously in the department for the isolation of RNA, है ना, और दूसरा herbicide that is based on the nanotechnology, lipid based nanoparticles adherent by nature which can be sprayed onto the crop, they adhere onto the surface of the crop and they are assimilated by the plant for the benefit and the eradication of all types of the diseases which they are suffering otherwise and afflicted of. हुआ, मैं चाहता हूँ कि अगली साल में मुझे यदि मैं जीवन में कुछ सुनने को रहे, सुखद रहेगा, शायद मुझे और मेरे शरीर के बीच का सेतु बनाने वाली सांसें यदि और आगे तक नहीं ईश्वर की कृपा से, तो इनका करने को प्रभु के कीर्तन से ज्यादा सुखद होगा कि मैं ये सुनूं कि हमारे विश्वविद्यालय के विद्यार्थी आज ट्रांसलेशनल रिसर्च की उन्होंने 10 MOU साइन किए हैं, या 10 लोगों को यंग साइंटिस्ट अवार्ड या क्या कौन सा है शांति स्वरूप भटनागर अवार्ड, लाइक ऐसी चीजें जब हमारे यहाँ आने लगेंगी, जब हम अच्छा करने लगेंगे तो जिम सुख संपत्ति बिना ही बुलाए ज्ञानशील पर जाएं सुहाए, अपने आप आएंगी। आप उनको अलंकृत करोगे, आपको अलंकृत होने की जरूरत नहीं होगी, ऐसी मेरी उम्मीद होगी। विद फैसिलिटी, विद अ माइंडसेट, बट द टोटल एम्बियंस दैट इज जनरेटेड ऑल अराउंड, आई थिंक very very bright future is there, radiant, repulsive, and that is sensibly it is pervasive by character is going to be there in coming future. With these words, I wish every success to this workshop, and once again I thank the organizer, coordinators, dean and director of DORD Thomas Shop, and dean of the School of Biological Sciences, and all the professors and my faculty fraternity friends sitting in here, that you just give me a very patientful listening. इस शब्दों के साथ कि मैं कुछ भी बोलता और मैंने अपने कानों में अंगुलियां डाली होती और मैं कुछ भी देना चाहता, तुमने हाथ बंद कर लिए होते, तो मैं असहाय से खड़ा रहता। तुमने बड़े ही धैर्य से मुझे सुन के मैं जानता हूँ ये सब चीज़ें तुम्हारे लिए बहुत ज़्यादा मायने नहीं रखती, क्योंकि आप पहले से इन चीज़ों को जानती हैं। लेकिन मज़ा तो वही है जो आपको प्यारा है, जो हमें प्यारा है, उसकी बार बार इसी को तो कीर्तन कहते आपने सुन के मुझे बड़ा कर दिया, इसके प्रति मैं आपकी व्यक्त करता हूँ। Once again I am thankful to the organizers for giving me this opportunity to be with their research. Thank you very much.
Thank you so much, sir, for your kind words. Now I would like to invite Professor Harold Thomas for the presidential address.
Thank you very much. Professor S.P. Vyas sir, Professor Varsha madam, Professor A.P. Mishra, Dean, Chemistry and Chemical Sciences, Professor from Medical College is with us right now, and all the eminent faculty members, car officers, and
our new librarian also, and dear the new workshop. The detail was given by Dr. Upadhyay, and later on a very meaningful lecture by Professor S.P. And from both lectures, we are really, I hope all the participants were very much motivated. Sir, I would like to mention one thing. Teacher is never retired. And our new faculty members, they always need your blessing and guidance. And whatever you have suggested, right now we have a sufficient high-end equipments, and as per the advice of Honorable Vice Chancellor Madam, I have circulated one letter to all the science faculty department, and I have received only one response from Dr. Upadhyay. Because as per the advice of Madam, at least 4 to 5 new centers we have to create for research point of view. Right now we have 8 centers, advanced research centers, that comes under CAR, and 5 more centers will be very soon, that will be start, based on whatever we are having high-end equipments in other departments. And as per your advice, naturally we will go for a separate bioscience equipments, and now is the time to joint work with medical science, with pharmacy, biotechnology, microbiology and zoology, so that good number of publication will be come out in near future. Right now also we have a good number of publication every year, but unfortunately we are not in, as per the NRF ranking, we are not in 100 within. And I hope with the effort of our Honorable Vice Chancellor Madam, within December, 88 new faculty member will join. And as per my knowledge, in biosciences and even in chemical sciences, the foreign faculties are maximum. If they all will join here in near future, so again we will achieve, or our flag will be again in high, like when you was as a student in 80s and before 80s, that time that Sagar University name was famous in all over India and even in abroad also. 2 faculty, especially that time, geology, applied geology and pharmacy, our alumni they have posted up to remarkable position in government organization as well as in industry also. And our alumni in almost all in different countries and India. And right now, I think in chemistry also same position, and biotechnology and other sciences also. So we are coming up very nicely in every field. And as per the guidance of our Honorable Vice Chancellor, we will achieve new positions, or we can say that we will achieve the heights in scientific field also. Sir, we need your blessing always. The blessing in informational sciences also. Your students like Professor Umesh Patil is here, then Kashav is here. Your blessing along with Professor S.K. Jain blessing and guidance is required for future research work in pharmaceutical sciences as well as in biotechnology also. I hope this workshop will be very useful for all faculty members and the participants. And another thing I would like to mention, as per the advice and guidance of our Honorable Vice Chancellor, all these workshops are organized without any financial support from university. That is on self-financing mode, and that is very much important because the chemical which we used in all these instruments are very costly. So, as per the advice of Madam, we have already organized 2 workshops that was also self-financing mode, and this is the 3rd. And within December or January, we will organize at least 5 more workshops on different equipments. I hope all the participants, they will interact with resource persons and they will apply that technique in their future research work. With these few words, once again, I would like to congratulate to Professor Varsha Sharma, convener of the workshop, and co-convener Professor C.P. Upadhyay, and all the faculty members of the biotechnology department and students, along with Dr. Rajkumar Koiri and other faculties, those who are involved in this workshop. My best wishes to all of you. Thank you very much.
Thank you, sir. Since we are at the end of this wonderful inaugural program, it is with great honor and privilege that we have the opportunity to felicitate our esteemed chief guest, Professor S.P. Vyas, who has honored us with his presence today. I would like to request Professor Harold Thomas to felicitate Professor Vyas with shawl, shriffle and memento as mark of our profound respect and heartfelt appreciation.
पूरे भाषण में एक चीज भूल गया मैं आए ना मुझसे पहली सी सूरत मांगे बहुत याद किया था
I would like to request Dr. C.P. Upadhyay to felicitate Professor Harold Thomas with memento. Finally, I would like to request Dr. Devanshi Upadhyay to felicitate Head of Biotechnology Department, Professor Varsha Sharma. I would like to request the organizing secretary, Dr. R.K. Koiri, for the vote of thanks to the gathering.
My warm greetings to all of you on the occasion of inaugural session of this 5-day national workshop come hands-on training on next generation sequencing and real-time PCR. Respected Professor Harold Thomas, sir, Honorable Vice Chancellor nominee, and at the same time he is our Director of Research and Development, Chief Coordinator, Center of Advanced Research, and Head Department of Geology. Respected Professor, yes sir, former Vice Chancellor Dr. Hari Singh Gaur Viswanathilaya, former Dean, Faculty of Technology, former Head Department of Pharmaceutical Science and Department of Biotechnology, and Chief Guest of this inaugural session. Respected Professor Varsha Sharma Madam, Dean School of Biological Sciences and Head Department of Biotechnology. Dear Chandrama Prakash Upadhyay sir, Organizing Secretary of this workshop. Dr. Talha sir, Dr. T.M. sir, Professor Umesh Patil sir, Dr. Vilasa Madam, Dr. Malvika Sikdar Madam, Dr. Sushil Kasav sir, and my respected dear fellow colleagues and friends, Dr. Neeraj, Dr. Yogesh Bhargav sir, and dear participants both offline and online of this workshop all over India and different parts of the world. Good afternoon and a very warm welcome to all of you from Sagar. It's a great honor and privilege to propose a vote of thanks on this memorable occasion. Let me first of all start by giving glory to the Almighty God and pray to make this 5 days national workshop a resounding success. This 5 days national workshop come hands-on training has been designed to provide participants with a comprehensive knowledge on the working of next generation sequencing, which is a DNA sequencing technology that allow researchers to quickly and cheaply sequence large amounts of DNA or RNA by simultaneously sequencing millions of small fragments. NGS has revolutionized the biological sciences by allowing researchers to study biological system at a level never before possible. Participants will also get an in-depth hands-on experience on real-time PCR, which is a major development of PCR technology that enables reliable detection and measurement of products generated during each cycle of PCR process. So after 5 days of rigorous and packed schedule with experts from both thermo and academia, it is expected that the workshop will definitely help the participants to enhance their knowledge and skills and train themselves on this new advanced technique. Further, we expect that this platform will provide the participants an opportunity to interact with themselves and know each other's area of work and foster collaboration in near future. So first and foremost, I would like to thank respected Professor Harold Thomas sir, Honorable Vice Chancellor nominee, and at the same time he is our Director of R&D and Chief Coordinator CAR, who despite his busy schedule found time to grace this occasion with his encouraging, inspiring words. We extend our heartfelt gratitude and thanks to Professor S.P. Vyas sir, former Vice Chancellor Dr. Hari Singh Gaur Viswanathilaya, former Dean, Faculty of Technology, former Head Department of Pharmaceutical Science and Department of Biotechnology, and Chief Guest of this inaugural session for accepting our invitation despite his busy schedule and enlightening and inspiring us with his wonderful speech that speaks a lot about his great personality and a great scholar. And the best part is that he's an amalgamation of both science and literature. And this is something that keeps on reflecting on each and every occasion we hear Professor Vyas sir. And a teacher in him with an urge to talk and teach. And we have on a different occasion we have heard him for hours. So that speaks a lot of volume about him. So thank you sir. On behalf of Department of Biotechnology and the organizing committee, I extend a hearty gratitude to Professor Varsha Sharma Madam, Dean School of Biological Sciences and Head Department of Biotechnology, and Professor Harold Thomas sir, Director of Research and Development and Chief Coordinator CAR. I extend my gratitude to convener of this workshop, Professor Emil Khan sir, for his guidance and motivation since the inception of the idea of organizing this workshop. I extend my gratitude to organizing secretary Dr. Chandrama Prakash Upadhyay, who was behind everything associated with this 5-day workshop right from its inception to its successful commencement. I extend my heartfelt gratitude to Thermo Fisher Scientific for sponsoring this event, and From Fisher team, Dr. Vishaldhar, Dr. Naveen, Mr. Ankur, and Dr. Arup Acharya, who is coming from Department of Geology, University of Allahabad, who has given their consent for and have shown active willingness to provide an in-depth hands-on training to the participants of this workshop. I also extend a hearty vote of thanks to our dynamic Center of Advanced Research team and technical coordinators Dr. Vivek Kumar Pandey and Mr. Ravichandra Prajapati and Dr. Dayanand Kori from Library Science for their active coordination of the event. I must thank the organizing team volunteers for working hard for the past 1 month. No program can be successful until and unless you get the support of your young, enthusiastic student team. So I am thankful to all the research scholars of the Department of Biotechnology, particularly Mr. Abhishek Pathak, Mr. Nitesh Mandal, Mr. Manish Maji, Mr. Kuldeep Gulaiya, who took all the pain from day one, right from creating Google Forms to designing certificates, workshop manual, and communicating to the participants. I must also thank Ms. Anjali Mishra for her active engagement and coordination of this event. At last, I congratulate and welcome all the participants who are coming from different parts of India for an enriched learning experience. Hope you all are going to have an enriched learning experience for the next 5 years celebration and hands-on training starting from today. So thank you everyone once again, with the hope that the event will be a great success. In the process, if I have missed someone's name unintentionally, I extend a heartfelt apology on my behalf. So thank you everyone and warm regards. Over to you.
I would like to invite you all for a short tea break arranged. Request all the attendees to gather back to the room since there is an scheduled lecture by expert from Thermo Fisher on basics and application of NGS. Thank you.
आकांक्षा बोलते आपने स्पॉट करके साथ ही आकांक्षा भी सर चलिए सर आप पहचान रहे हैं साहब आइए आइए
कह रही है हमें लगा कौन है ये क्या तो ऐसे क्यों पकड़े
NGS इसमें बोलो बोल दिया कैमरे में
कैमरे पे
ये
अभी
स्ट्रीम
उसी
थोड़ा
टाइम पे
ये बोल दिया ये बोल दिया कि बड़ी देर से स्टार्ट करें आफ्टर हाइट ये कैमरा है भाई
या Lecture on basics and application of NGS will be streamed sooner after a short break.